pleasure to give talks on today, this afternoon again, and then also on tomorrow and on Saturday. And at the end, if you follow all the lessons, you will see how it belongs together. There is some connection in between. But in fact, breeding for resistance is maybe my personal main topic. I work on this since I started in bee research activities 30 years ago. Still, the problem is not solved. So you see already it's not an easy game. But this has something to do also with the way how we keep our bees, how we manage the bees. So beekeeping techniques and breeding has to go hand in hand. And this is one of the main messages already. But let me start with what do we know about resistance? And we are lucky. We got the mite from Asian bees, from Apis cerana, and those bees do very well with Varroa. So we have a living example how resistance can work. The difficult thing is it's a different species, so we don't have a chance to directly cross cerana with mellifera bees. That would be maybe the easiest way to introduce some traits which we would like to have in our mellifera bees. But at least we can learn from them. And they have a very similar living way of living. So nest bees with parallel combs, with more or less identical inner brood nest conditions, humidity, temperature, and so on. And this is the background why this highly specific parasite called Varroa were able to switch over. And when we look for the details, what those bees do with Varroa, first of all, we find an intensive grooming activity of the infested bees and of nest mates. And then you can easily find mites which are hurt and killed by the bites of the bees, as you see here on the picture. Furthermore, when you have a closer look on the brood, you find brood cells like this, where there's a pupae developing and there's a mite in, and the bees react. They find out those cells, they open it and clean it. So brood hygiene behavior plays an important role. But nevertheless, it's a rare situation in cerana that Varroa invades worker broods. So they mainly go to drone brood. And this is a close picture of drone brood in cerana. So you see, it looks quite similar to what you know how drone brood looks in mellifera. But still, there is one curious thing with these holes here in the elder cells. You don't have it with the freshly sealed cells. Those holes develop later on by an enzyme which is produced from the pupae. And they need to have this hole in the cap, because the cap is so dense. So the cocoon is much harder than it's in mellifera. It's so closed, well closed, that there wouldn't be sufficient oxygen in the cell for the pupae to survive. So that's why they do this hole. And this is linked to Varroa. So if Varroa goes in such a cell and the cell is kept, then it's really protected, because the sealing is so strong that the bees from outside wouldn't open it again. So this is how you have the difference between worker brood and drone brood in cerana, and that makes drone brood as the only host for Varroa in those colonies. And the next thing what happens is indicated by those black cells here. If there are 2 or more mites invading such a drone pupae, then it can't survive. It will die. And at the moment where it dies, the cell remains. And we don't know this from mellifera. Our bees are much more clean. So if a cell dies inside the hive, it's soon opened and cleaned out, and then the mite is still alive. But if those cells remain over a week or longer, then all the mites inside the cell are killed. So it's like a perfect control that you know that never have an overdevelopment of Varroa mites in those cerana hives. So there are some further traits, but the most important are listed here. It's this active grooming of adult bees. It's relative unattractiveness of worker brood, And if mites invade, they are easily detected and the brood is removed. Then mites can only reproduce in drone brood, and you have a seasonal occurrence of drone brood as we have it in mellifera. If there are too many mites in, then you have this entombing of parasitized cells, and the mites will die inside the brood. And last not least, if there are too many parasites in such a hive, the bees get nervous and they abscond, a behavior which is also known from mellifera, but it's very rare. So we have a swarming as a part of the reproduction system of honeybees, but we don't know so much about this absconding, which means all the bees, together with the queen, just leave the brood nest, leave back honey in the former nest, and all the bees are gone from one day to the other. That's what Serrana does at the moment where there are too many parasites or other disturbances given. I made some bad experience, by the way. Just to tell you, I had a chance to work with Serrana some time ago, and then we wanted to measure the post-capping stage duration. So the way was we opened the colony every few hours to take pictures, and with most of the colonies, after the third or fourth time when we came, every 4 hours, all the bees were gone already. That was too much for them. So that's about Serrana. Now we come to mellifera, and there again we are quite lucky because in the meantime we know a number of highly resistant populations of our European bees. In Europe, in United States, at the end all over the globe. So there is no doubt any longer that higher resistance level can easily develop within mellifera. The way is, how do we reach this, and on which conditions does it depend? And it's an interesting and curious point that all those populations which we know developed from natural selection. So the bees were left alone with these parasites, and it took some time, but then at the end colonies occurred which could manage with it. That's great, because those bees can better show us what is changing, what are the differences, what do we have to look for. And one crucial thing is what Yves Leconte from this resistant population in south of France, in Avignon, showed some years ago. It's published here in Apidology. So he compared the resistant stock with commercial stock from the neighborhood, which was at least once a year treated with amitraz. And you see this here, this is showing the natural mite mortality of those hives as an indicator of the mite population development. It's going over 3 seasons. And you see they had a treatment in the commercial stock to bring it down, but then in the next season it went up again, and they had to treat again, and it went up again, and they had to treat again, while the resistant stock remained on a comparatively low mite infestation level all the year around. And this is already going on since 15 years. He's observing this. So it's quite stable. So mite population dynamic is different in the resistant stock. The next thing which we know by now is if you check the reproductive success of Varroa mites which invented worker brood, there you find a difference. And this is shown here for this resistant goatland population in Sweden, and again for the Avignon population. And while susceptible stock from the neighborhood shows 80 to 90% of success in reproduction of mites in the worker brood, it's going down to 50 to 60% in this resistant stock. So about half of the mites invade the cells, but at the end, when it comes to the brood emergence, you don't find adult female offspring of those mites for different reasons. But this seems to be a very clear change, and we find this in most of the resistant populations which we can follow now around the globe. There is one more thing which came up recently, and it was published here with my colleague Melissa Oddy, and we contributed with our data. And this tells the story that even if the bees don't remove the brood, but just detect infested cells, open them, and then later recap them again, this has a negative effect on the development of the mites inside the cells. We call this recapping behavior. And this is relatively new, but we see it in all the resistant populations. Again, you have here the bees from Avignon, and there is a second resistant population in France. We also know, meanwhile, a population in Norway which has a higher resistance, and there are these goatland bees again. So we could include samples from all those populations, and we again compared the surviving colonies with colonies from the neighborhood which are susceptible for mites, and you see this huge difference. Here it's more than 60% of the infested worker cells, which were at least once opened and recapped by the bees during the brood development. And it's just about 20% in the susceptible stock. And you see this difference is for all those populations. And we also compared cells which were infested and cells which were non-infested here on the right. And again, you have the difference between the resistant and the susceptible stock, but in general, it's on a much lower level. So there is a directed reaction on the infested cells. Here in this nice but complex graph, we try to bring together all the things which we know by now on resistance. And I'm sorry about the people sitting in the back. I checked this graph this morning and noticed these small written things here you probably can't read. I shortly explain the situation because it's really crucial for us to understand resistance is not one single character which has to be changed by breeding, and then you have queens which you can introduce into your hives, and whatever you do with those hives and wherever you keep them, they are resistant. This is not the truth. It's much more complex. First of all, what the bees do inside the hive is always connected to the environmental conditions. So we have these factors here. It's about nutrition. It's about climatic differences. It's about the density of the bee population around. And last not least, it's a beekeeper who has a main effect on what is developing inside the hives. So there are many, many factors, and once it comes to a resistance in a specific population, it's always about adaptation to the environmental conditions. And this afternoon talk will be very much about this interaction of bees and environment. So what is adaptation, local adaptation? It plays a crucial role. We know this by now because, for example, we tested bees from Goatland, we tested bees from Avignon and other resistant population in different places. We got them, for example, at the Institute in Kirchhain, and it didn't work at all. And at the same time, the sister queens from these test groups worked well in Goatland and worked well in Avignon. So this is, first of all, to note, you can't import the bees from somewhere. You have to develop them on your spot, under your conditions, and with the way how you keep your bees. And then bees come into the hive, what we call Varroa invasion, always sitting on bees, but at the same time, mites leave the hive sitting on bees, and they are lost in the field or they move to other hives. So this is in and out, and this has very much to do with the density of colonies and with the flight activity and with the ability of bees to orientate in the field. So once the bees are inside the hive, they are on the adult bees, and they try to go into the brood. And they come out from the brood again and back on the bees. So with sitting on the bees, of course, we have this grooming behavior. So the bees always try to find the mites and to fight them, and they can kill them. That also happens in your hives at home. A certain percentage of mites is lost by grooming. So they go down, and this affects the mite population development, which is staying down here. More interesting is what happens inside the brood. And there are certain characters. So we know brood can be more or less attractive for mites. There are certain chemical stimulus in the game. Or for example, there is this differences in post-capping stage duration, which affect the number of mites developing within the cell until the brood emerges. So we have several characters which are bound to the brood. But the most important things when it comes to resistance are staying here, and it's behavioral traits. And there is this brood hygiene behavior, that the bees are capable to detect infested cells and then to open them. And one way is what we call VSH, Varroa sensitive hygiene, that they clean out those cells. So they definitely stop the chance for the mites to reproduce in this specific cell because they take out the cell. What happens to the mite is that at that moment, she usually survives the act. It's rare that the mites are killed during VSH actions, but the mites escape, they are back on the bees, and they try to invade the next cell as soon as possible. So the circle is going on. But probably the mite already laid some eggs, and now it's back on the bees, so the normal reproduction is disturbed. It can introduce the next cell, but then maybe she goes on with egg laying because she is highly pregnant and the egg is already ripening and has to be laid down. So in the next cell, you get a disturbed, non-synchronized offspring compared to the bee development. So this is a crucial disturbance. You don't kill the mites, but you disturb the reproductive behavior. And that at the end affects the mite reproduction. And then there's also this recapping behavior. And till now we do not know in the final detail how it works, but maybe there's a similar effect. We know, for example, at the moment where cells are open, sometimes mites leave the cell. But at the same time, when these cells are open, mites which are on the bees may go in. It's not only the cell which is close to the first capping 9 days after egg laying, which is attractive to the mites. It's also these elder cells which are temporarily opened through this opening and recapping behavior. So it's a complex activity, but this is probably the focus of resistance in mellifera bees, these activities here. And at the end, it all affects mite reproduction. So the ability to lay eggs in general, and then the number of eggs laid. And mite offspring can die in the cell for certain reasons. And at the end, the young females have to get successfully mated by its own brother in the cell to be fertile afterwards. So many aspects on mite reproduction. And this is what we call SMR, suppressed mite reproduction, that in resistant colonies we see it's so much disturbed that only a limited number of mites can successfully reproduce. And that all those things affect then how mites develop inside the hive, so we can measure this mite population development. So you see, I have these bold letters on some of the traits. So it's grooming, it's mite reproduction, SMR, mite population development, and then here this hygiene behavior, VSH, and recapping. And probably those are the traits which are most interesting for further breeding. This is also indicated by this questionnaire. At the moment we are running a European study. I come back to this in a few minutes. And as part of this study for the European Commission, we asked experts around the world, so 48 experts from Europe and from North America, Canada and US, about which traits do you think are important for the selection of resistance? And this was the outcome. So there 7 people answered, well, colony survival. Don't affect the hives, don't treat them, and follow which ones do survive over several seasons maybe. Mite infestation, one of the most important characters. So check regularly how mite population develop inside the hives. Hygiene behavior, in the sense of this general hygiene, pin test or frozen brood test, might be useful. Then SMR, so checking how many mites have successfully reproduced. VSH, which I just explained, recapping. And then some people think grooming is important. Some people say it's mainly the colony strength, so just go for the strongest hives. And there were also some curious other things mentioned as potential characters for selection. I think this gives a very clear picture worldwide how experts nowadays, with all the information which was collected throughout the last 30 years, think about our potential for resistant selection. What are we doing at the Institute in Kirchhain? So I come now to our specific work. And you have to know this Kirchhain Institute among the German bee institutes is the one which has the longest tradition and most efforts in selection and breeding, maybe together with the colleagues from Hohen Neuendorf, Caspar Bienefeld. So we closely cooperate, but it's mainly Kirchhain. So we have several institutes and each institute has a different focus. Our focus is on breeding. So we developed testing protocols already a long time ago when it was more about honey production and gentleness and all those things. And now we are so much involved in this selection on resistance. So what we do for this is we usually start about 60 colonies. This year it was 80. And we build them as shook swarms. And they have a very low Varroa infestation at that time. So we build them beginning of July. And then we use only pure mated queens. And in this specific population, it's a pure Carnica population. Pure mated queens means we know exactly the pedigree. And we rear the queens and we bring them to isolated island mating stations, or we use artificial insemination. Both is well established. So we know exactly the genetics standing behind. And then we follow the course of Varroa development in those hives, but we don't treat them. We start them in July, and then we get bee samples already in November to see how many mites are there now before winter. And then next season, we take several bee samples in June and July to see what is going on there. And then we run a 1-year performance testing, including, of course, productivity, gentleness, very important. We want to have a low swarming tendency. We want to have strong colonies. All those things are measured. And included in this routine performance testing is the so-called pin test for brood hygiene. And then when it comes to the end of the testing period, usually we collect the last honey about the mid of July. So at the end of June, we go to cage the queens. We want to stop brood production. And then 4 weeks later, when all the brood is gone, we have a single treatment with oxalic acid, which works very effectively, which kills us all the mites. So we have a good impression on the final mite propagation after 1 year without any treatment. So it's really a full season. And shortly before the last brood is gone, we take 1 comb of brood, and this is then in the lab studied for SMR and recapping. So I will show you the methods. This is how we sample the bees for Varroa control. We always take bees from an outer comb 40 to 50 grams of bees. So it's these small plastic containers taking 120 milliliters. And we bring them home. The bees are already dead then. And then we wash them with soapy water. We shake them about 15 minutes. And then you put them over such a screen, wash them, and all the bees are kept back, and the mites go to the finer net, and then it's easy to count those mites. And then you know you had so many grams of bees, and there were so many mites sitting on. A very reliable and simple working method to identify the recent infestation level of the bees. About the pin test, I don't know if you are familiar with this. It's well established meanwhile. So you just take a brood comb with preferably younger pupae, so white eyes or reddish eyes of the pupae, and then you choose such a part of brood, and with a fine needle you pierce 50 cells, row by row, and you mark the upper left corner and the lower right corner, and then the cell 51, so that when you come back to the sample some hours later, you know exactly here all those cells were treated with this fine insect pin, which in a way should, how to say it in English, it's a bit similar to what mites does. They pierce the pupae. We don't kill the pupae with this pin. It's just to hurt the mites, because we know it's a hurt pupae which sends out the signals for opening the cells and for this hygiene behavior. So, and then some time ago we had longer intervals. We used 8 to 9 hours. Since last season, we check those cells after 6 hours, and then it looks like this. There are still some cells remaining which were treated, and the bees didn't find them, so we count how many cells are still untouched. And then we check from the cells which are opened, is there still brood in, or is it completely gone? So is it completely removed? This is what you count then 6 hours later. So it takes you about 5 minutes per hive. It's a simple test, but well standardized, and it works very, very nice. And then finally, this brood comb which goes to the lab. We store it first in the fridge, and this is work then in the winter time, because it's really time consuming. And this is my colleague in our lab who does a great job. So she spends meanwhile about 50% of her working time evaluating such brood combs, which we get from our own hives, but also from participating beekeepers in the breeding program. It takes you 2 to 4 hours for one sample, because you have to go for at least about 25 single infested cells to get a reliable result. And it depends very much on the infestation level. So this is why we take the samples shortly before the final treatment of the hives. So we would like to have about 5 or 10% of the cells infested with Varroa. Otherwise, you open thousands of cells and don't find mites. You just lose your time. And what we do then, when we open the cells, first of all, we carefully open the cap. We cut it from the edge, and then we try to turn it around so that we get a view on the inner side of the cap, because this tells us about recapping activity. And recapping looks like this in your hive. When you take out combs in summer time and carefully check, you will from time to time find cells like this, where there is a pupae in, and it looks healthy, nothing special about it, but the cap is gone. Why? And mark such cells, put it back, and go again a few hours later, and then most probably the cell is recapped again. So you can directly observe this activity. And it was long time overlooked. We always thought, like as it is written in the books, capping after 9 days, the pupa is developing, and then on day 21, the young bee is emerging. Meanwhile, we know from a student work who did video recording of such combs, it can be often opened and recapped again in this interval. The extreme value which we had from this video observation was one cell 17 times opened and closed again. Maybe a bit artificial in such an observation hive, but it clearly shows it's a frequent activity. So the bees are always communicating with their brood, and they are active. Interesting. So you can directly observe this. What we do when we have these frozen brood combs, we carefully open the capping, and this is how it looks like if the cell was not touched by the bees before. Then you find on the inner side parts of the cocoon, this silky white shiny material, like here. But if the cell was open, completely open like this one, and closed again, then it's only a wax layer without this cocoon. And you see the difference in the color. But often you find cells where it was just a small spot opened and closed again. Then it looks like this. So it can be done. And then when it comes to suppressed mite reproduction, first of all, you have to go for elder pupae. The youngest stage which we use is when you have these purple eyes. White eyes or red eyes is too young. It starts with purple eyes, and then later on the eyes get black, and then the whole body is colored afterwards. So this is the youngest stage which you can use. And for those cells, you check then if there is a single mother invading such a cell. At that stage, you can expect that there is the eldest daughter already in this so-called deutonymph stage, which is the same size and shape like the mother, but still white. And there is a son running around because this is the eldest offspring from this mother. So this is the son. He's one day elder than this deutonymph, but still in the deutonymph stage. And there is the next protonymph. This is the second eldest daughter. So this is a normal reproduction activity at that age of the brood. But at the same time, you find mites like this. There is the mother mite, and there's also remains from the shit of the mite, so she was active in the cell. But all we find is a single egg here, much too young to develop into adult offspring at the moment where the bee will emerge. So this is a difference. And there is a clear protocol meanwhile. So basically what you do is you identify the age of the brood by these color marks down here. So with some experience, it's not a problem to say, well, this is 7 days, 8 days, 9 days after capping. And then you compare it with what you see from the mites. You always expect to find the mother mite. And when it comes to the males, in this early phase, you still have protonyms, and then later on you have an adult male. And then you expect the eldest daughter to be a deutonymph, or from day 10 onward, there should be already an adult brownish daughter mite. So this is the principle here. And there is a clear protocol now how people work. Use only cells where there is a single foundress. If you have multiple infestation, it's an infested cell. You count this. You want to know about the brood infestation level, but you can't evaluate the reproductive capacity. And then you check for the age of the brood, and if it's 7 to 9 days, then you expect a deutonymph and a male present, and then it's normal reproductive. But if the male is missing, or if the eldest daughter is still in a younger stage, then it's non-reproductive. And if the brood is elder, 10 to 12 days, then again you expect here adult daughter and an adult male present, then it's normally reproductive. If something is missing, it's non-reproductive. This is how we work. So now some data from this research. And this, first of all, is how it looks like with the Varroa infestation development in our test population. Here's mixed data from, it's a data from one season, but it looks quite similar from year to year. So you see we have a reasonable infestation in the winter hive. So starting with nearly 0 mites in July, we often get about 1 to 2% Varroa infestation in the winter bees. if we don't treat at all, as we do it here. But there are already some colonies which really go up to 5%, which means with 10, 15,000 bees in the hive at that time, you have here 500, 800 mites maybe, and we don't treat them. We are not worried about it. We have healthy bees, and they can well live with some of those mites. So that's how we start the testing, and then you see, in June it's still this low level, but the number of mites went up, but they are all now inside the brood, and there's plenty of brood at that time. But the infestation is then growing, and when we take the final sample in the mid of July, the queen is already caged, so most of the brood is gone. And then, of course, all the bees go to them, all the mites go to the bees, and the infestation is going up. But now you see the advantage. This is not commercial honey producing, but it's performance testing. We want to see the differences between the hives, and all got the same starting conditions, and see the huge differences which we find in Varroa infestation. You have the same with your hives, but probably you don't recognize it. You are not aware that there are some colonies who really have a problem with Varroa, while others do very well. Here we see it clearly. Colonies which still remain with less than 5% mites on the bees after 1 year without any treatment. Excellent stock, while other colonies have a huge problem at that time. So this is testing, and this is selection. When it comes to the level of different sister groups, so genetic comparison, then you see that there are certain lines which really have a problem. All those sisters here in this group had 10% or even higher Varroa infestation in June, July, while other groups like this one had no problem at all. All the sisters were fine. So this is where we are standing. There are huge differences. We have to find them, and we have to select for it. All of those colonies were highly productive, gentle stock, no swarming. Everything is excellent with those bees. So this is evaluation of the pin test here. In this case, it's from previous years, so it was still after 9 hours, but it doesn't change too much if you come down to 6 hours. What you see here, this is in non-open cells. So it's not so many cells which remain untouched, but we find colonies which still have 20, 30, or even more cells non-detected after this pinning. And those colonies regularly have a big problem with Varroa at the end of the season. So we want to find them. This is a negative selection. All the colonies should be able, within 6 hours, to detect cells which were clearly hurt with the pin. And this is about the total removal. And you see it's about 50% of the cells are already completely gone, but some colonies nearly 100 after such a period, while other colonies have 20, 30% removed. So there are, again, interesting differences. When it comes to the recapping activity, there are even bigger differences. And this is recapping. 0% recapped, 100% recapped. We find all these differences within our population right now. But there are many, many colonies who do not detect Varroa-infested cells. They don't touch them. They don't open and recap those cells. So, but there are other colonies which find 100% of the Varroa-infested cells. Very crucial point, not too difficult to measure. And because it's easy to measure, we already started to select on this trait, and this is how it developed. Season 16, 17, 18, you see it clearly goes up. It clearly goes up. This bold line here indicates the median colony, and you see, meanwhile, we have about 45% of the cells recapped in our population. So it's relatively easy to select for. And this is about SMR. Maybe at the end, the most important indicator. So are there how many mites without reproduction, and this is listed here. So what we think is normal in European colonies is something between 0 to 20%. All those colonies here, this is what usually happens. Some of the mites fail, but there is no indication that it happens to many mites. Interesting for us to see that we find colonies where we have 30, 40, and even higher percentage of non-reproductive mites. And when we go back to these resistant populations, which I showed in the beginning of my talk, you remember it was about 50, 60% in those populations. So we find some colonies which reach a similar extent. Probably we are not so far away from this, but it's extremely difficult to get this stabilized in the population. But it's because it's affected from so many things, even environmental conditions. It's not just a genetic trait. So it's hard to select for. And there is some increase over the seasons. So we started in 2013 to measure all our colonies. Here you have the number of colonies and the average values. And by now we reached a level of about 30, 35% on average. But there's not such a steep increase as I could show you for the recapping. It's still some work ahead, but I think it's an important selection line. And right now, this season, the National Ministry of Agriculture in Germany started a support program for breeders who join this kind of selection. And we are coordinating this from the institute in Kirchhain, but there are quite a number of Carnica and Backfast breeders who are working on this now. So we hope to make good progress on this character within the next few years. And to better understand all the background, to be honest. We do not understand the whole biology which is standing behind SMR. Still many questions about this. But extremely interesting also from a biological point of view. So when I put all the things together, we see correlation between the characters. And finally, this mite population growth is the most important things. If the mite population doesn't grow so much during the season, then you don't have to treat. So this is probably what we would like to call a resistance. The precise definition for resistance from the biological point of view is survivability over long periods without treatment. We don't measure this, then we would have a high risk to lose colonies, and it's routine testing. But we can measure this mite population growth. And if there's a low population growth, then we know those colonies have a high chance to survive over years without treatment. So this is why we relate the other characters to here. And then when we have these pin test results, and we see at the end of the season, there is a highly significant correlation. So colonies which have more cells open in the pin test have less Varroa mites at the end of the season. It's a correlation of close to 0.3, highly significant. There is this recapping of infested cells. So how well do the bees detect infested cells? And if they do well, then they have less mites. It's a lower correlation, 0.14, but it's negative, which is good. And there is a weak significance for this. So recapping directly affects this trait. And then there's SMR. And again, we have a similar correlation like for recapping. Colonies which have less reproductive mites have higher mite loads at the end of the season. So it works as expected. And interestingly, there are correlation between those traits. So when the colonies open more cells, they also show a higher recapping of the infested cells. And if they have a higher recapping, then they have a higher SMR as well. This is good because it fits into the model, and that means for the way of testing, when we measure one thing and measure the other at the same time, they strengthen each other. So we get a clearer selection decision at the end of the year. Well, I'm coming to the end of my talk, and it's a bit like an outlook, but it's a recent activity, and it's great to see this. The European Commission, forced by a decision of the European Parliament, decided 2 years ago to run a study. They want to know what is the potential of selecting honeybees, European honeybees on resistance, what is already available, which techniques can be recommended, which kind of support should be given to the industry to speed up with the resistance selection. So that's why they called us for a study, and there was like a competition, and we won this competition. So I'm the happy one who is now in charge to coordinate this European study. This is our institute here in Kirchhain, and we closely cooperate with the colleagues from France. This is INRA, and CREA is the respective board in Italy. So these are our closest partners, and there is an administrative company involved, which is also located in Germany. This is a core team which organizes the study with the help of many, many experts around, also from Britain. Norman Carrick, for example, is part of our scientific board and is supporting this study. And these are the main tasks. So first of all, make an analysis of the European market for breeding stock. So how many queens are produced, how is about import, exports, all those things. This was already done last year. We are not allowed to publish those data because the study, you're producing data for the one who pays you. It belongs to the European Commission. But of course, at the end, they want to extract the essence and make their decisions on it. And there will be a large conference in April 21 in Brussels, where all those data and conclusions are presented to the field. And from then on, those things will be available. The next step was, what is the status quo of selection? And the questionnaire, which I showed you before, is coming from this part of the study. So we asked experts, and we checked, are there any resistant populations according to the reports of beekeepers and breeders and so on. And now we are in the core part of the study, and this is to run large-scale field testing on these assumed resistant lines, which we have right now. And this was started this summer. So the queens are now in the hand of many, many testers around Europe. And we start to collect data. In these days, they have to check for the strength of the colonies before wintering and the infestation level. And then next year, they will collect many, many data. And we will put all those things together until the end of next year, and then present it in this final conference. So this is how the decision was made. You can't run this study in 27 member countries. We had to make a selection, and it's always discussed with the steering group of the European Commission. Every few months I'm in Brussels, so I feel home meanwhile there. And we decided for France, for Italy, Greece, Poland, and then finally Germany. And with the German study, we linked it with Austria and also Croatia, because there's a lot of exchange of breeding stock, carnica breeding stock going on between those countries. So these 5 case studies, and we mark here the source of queens. So for each case study, we have at least 3 different lines assumed to have already some resistance from natural selection or from selective breeding of people. And we marked also the performance test yards, because each of those 3 lines is been tested in at least 3 different performance apiaries, performance test apiaries, and performance test apiary mean they collect all the data, all the economically important data, but also all these resistant traits, including recapping and SMR and all this. So it's a huge workload, you can imagine. But maybe more important, because it's about the use of such resistant stock for commercial honey production in Europe. This is the idea of the Commission. We involved many commercial beekeepers. And in their case, they simply compare one line of our resistant stock to the bees which they usually have in their enterprise. So it's just one to the other. And the way how they compare, they got the queens, and they should manage their hive as they usually do under commercial conditions. So they move them, they want to produce a lot of honey, they don't have time to do many specific measurements on the hive, but they have to make an income from this way of beekeeping. And they will tell us at the end, are those bees suitable for commercial honey production? So can you easily deal with them? Do they survive well? But they will also measure Varroa infestation development in those hives. So that's the way of the study. And the extent is impressive and give me some sleepless nights, as you can imagine. So this is the setup, how many genotypes went into those different studies, 22 altogether. This is about the races. You see we have Carnica, we have Buckfast, we have Macedonian bees, we have some local stock. We have mellifera bees we have Siciliana bees and Ligustica bees. And there's some link between the studies, so we have also some international exchange, but it's mainly going with those lines which come from those countries. And here's some information about the main traits which were regarded in the previous selection. So this is the general hygiene behavior, SMR, this is survival, this is VSH, here with some of the groups. So that's what it's all about. And this is a number of queens which were planned to be exchanged and distributed. So it's together more than 2,000 queens. And de facto, I can say now we even reached higher numbers. So we had some spare stock to be on the safe side. And I think we distributed about 2,500 pure mated queens last summer. It's now in the hand of the beekeepers, so curious to see how it works, and pretty soon we will have a much better information. Is there really something which we can make call resistance, or do we at least identify certain selection strategies which are worth to be better supported by the European Commission? And hopefully we have some positive results in this to ensure that this, I think on a longer term, extremely important perspective to get resistant stock, to get a chance to no longer use chemicals year by year in our hive is so important that we should go in this direction. And in this sense, let me thank you first of all for your attention, great audience. I was surprised that it's so crowded this morning, early morning, and now we have chance to discuss things.
Is there any evidence for a genetic component in the mite genetics in the suppressed reproduction? In other words, are they shifting their genetics, or is it entirely the bee behaviour and the management of the bees that are creating the suppressed mite reproduction?
So the general situation with Varroa is that there is a very, very low genetic variability within the population. And this comes due to the mating biology. They have inbred lines. Sometimes it's like a clone. Still, there is some genetic variation, and with modern genetic tools, we can now better identify it. You know it, for example, from the development of resistance to certain drugs. So there is a slight genetic change in such mites. And that gives a chance that also differences between the mites may play a role, but it's for sure less important than what is done by the bees. More interesting is the biography, let me say, of the individual mite. So it can be affected by viruses, for example, or rickettsia. So also the mite can get ill. And as I already discussed, if mites are released from a cell at the wrong moment and have to go to the next cell, then they are disturbed with this synchronization. So facts like this are on the mite side and affect SMR. Yeah.
You mentioned you have healthy hives that you're working with, and they're living with the fairly strong mite population, it sounds like, and coping with it. And you mentioned the changes of genetics possibly in the mites. When I look at my hives, and I see something like parasitic mite syndrome or these other vectored diseases, are your mites healthier than ours? Or, I mean, what are we sort of looking at in the mite spread of disease, so to speak?
I'm not sure if I got all your points. Sorry.
So to simplify that, are your mites healthier than our mites? Or my mites, you know? Because you said your bees were staying healthy, sustaining a mite population. Mm. But, you know, my bees this summer were doing very healthy. But I've come to find this autumn that they're taking a crash. Mm. And, you know, so they weren't maintaining this healthiness. Mm.
I don't expect that there are significant differences between our mite populations. We have no indication for this. It's all Varroa destructor, and as I said, there's, from all the studies we know, very, very low genetic variability between those mite populations, which spread over Europe and North America. There might be important differences when it comes to the interaction with viruses. So this is a critical point. When we lose colonies by Varroa, it's not Varroa at the end, it's the viruses which kills the bees. And earlier or later, you always have this sharp build-up of virus infections at the moment where you have higher Varroa loads in the hives. And there we know that there are important regional differences. So sometimes it goes together with deformed wing virus, other places it goes together with slow paralysis virus, and so on. I think you are informed on this, that there are plenty of mites viruses in the game, and we have different interactions with Varroa. So that can make a huge difference, and you always have to be aware of what are the virus loads in my population and how is it developing. And there's even this discussion now about different genotypes for deformed wing virus. You know, we have this type A and this type B, and type B is much more virulent than its type A. But what we know by now from the studies in Germany is that we always find B, so we have the most critical one. We have strong hives, and at the end it's always a question of the relative infestation levels. So this is our indicator. How many % of the brood is infested? And I think I come back to this when I talk on Varroa on tomorrow. That you will better understand maybe. And we can easily go with no winter treatment as long as we are sure that in August, September, at the time when the winter bee population is reared in the hives, we don't exceed certain infestation levels. So there is a huge misunderstanding in the wider bee community. You all learned that you have to do a winter treatment. But this is maybe the biggest mistake we do all over. We don't need a winter treatment. We need a treatment right in time before we produce winter bees. And this is July, early August. And if you come down with your mite load at that time, then you will have healthy winter bees, and they can easily bear a few hundred mites, and then you have a good chance for the next season to see the differences between the hives. What we do at the moment, that we kill all our mites in December, just cut out any option for us to select for the differences which could develop then during springtime.